Alright, this week we are going to study generalized connective tissue and cartilage in the lab. This is a section of mesenchyme. Mesenchyme is an embryonic type of loose connective tissue. The particular slide where you find the mesenchyme will have other tissues that have already developed or these cells have differentiated already into some other tissues. So you're going to have to look to find this among other tissues. And mesenchyme tends to be one of the most unremarkable parts of the slide. The cells are not packed very densely together. They have kind of a very regular oval-shaped appearance. They're slightly elongated, and the cells have lots of fibrous extensions or cytoplasmic extensions. There are no extracellular fibers in the extracellular matrix in this tissue, so these long processes you're seeing here are not fibers, rather they are cytoplasmic extensions of the cells themselves. And so these cytoplasmic processes possess lots of chemoreceptors. The cells are actively sensing chemical gradients which may allow them to differentiate into specific types of tissues. So this is mesenchyme. You may also see some developing bone or blood vessels or developed types of connective tissues on that slide. This is a slide showing mucus tissue or mucoid connective tissue. This is another embryonic loose connective tissue. This cell actually contains fibroblasts. So in mesenchyme, the cells are called mesenchymal cells. Here they have differentiated into fibroblasts. This particular slide stains the collagen fibers in the matrix a bluish coloration, so these are actual fibers in the matrix here that are staining blue, and a counter stain has been used to stain the fibroblast cells a reddish color. So there may be other fibers in the matrix as well, but collagen is the main type of extracellular fiber that's visible in this slide. Here's another section of mucoid connective tissue where it's a little bit higher density. So you'll have to look around on the slide. This particular type of connective tissue is only found in the umbilical cord. It's sometimes referred to as Wharton's jelly as well. There are blood vessels on the slide, and those tend to be the most eye-catching, prominent structures. So usually you can find the mucoid tissue surrounding the blood vessels, so look a bit outside of it. This is a section of areolar connective tissue. Areolar connective tissue is the first adult loose connective tissue that we'll look at, and it's the most ubiquitous loose connective tissue. You can see the nuclei of many cells here. Most of these are fibroblasts, but there may be other types of cells, including macrophages or plasma cells, that are commonly found in connective tissue. Most of them are fibroblasts, but because you can't see the cytoplasm of the cells really well, it's very tough to distinguish between cells just based on the appearance of their nuclei. So assume that most of these are going to be fibroblasts. You can also see two major components of the extracellular matrix here. There are two major fibers. You have the collagen fibers, which in this case, because this tissue has been stained with H&E, the type 1 collagen fibers here, these very thick fibers, stain eosinophilic. If you go back in this slide, this is also type 1 collagen, but it stains bluish because it's a different type of stain. You don't need to know the particular stain that was used in that slide. So with H&E though, type 1 collagen appears pinkish. Additionally, you can see some other thinner fibers that stain more basophilic, giving it a kind of bluish black color. These fibers are thinner, and there aren't as many of them. These are called elastic fibers. So you can see several elastic fibers in the slide. There's one here, here's another one, here's one, here's another one here. Elastic fibers get their name because of their elastic properties. So the more of these types of fibers you have in a tissue, the more stretchable or elastic it tends to be. Here's another section, a little bit higher magnification. Mostly you're seeing the type 1 collagen fibers here. Here's a single elastic fiber that's kind of coiled around, kind of looks like a little thread or hair here. In areolar connective tissue, there's actually a third type of fiber, which are called reticular fibers. Visualizing reticular fibers requires a specific type of stain to be able to see them. So we'll look at some tissues here in a little bit that have been stained with a silver stain, so you can actually see those. So know that areolar connective tissue has all three fiber types: elastic, type 1 collagen, and reticular connective tissue, or reticular fibers rather, but you can only see two of those when stained with H&E. This is a section of adipose tissue. Adipose tissue is actually classified as a type of loose connective tissue. The most obvious component of adipose tissue are the large spherical shaped adipocytes, which most of their cytoplasm is filled with lipid inclusions, so they mostly contain fat. This particular tissue section has been stained with osmium tetroxide, which, as we've seen before, stains the adipose cells black. But in addition to the adipocytes, there are other cells that can be found. You can find fibroblasts and other immune cells. You can also see collagen fibers in the extracellular matrix here. This is tissue that has not been stained with the OSO4. This is adipose tissue still. And you can see a lot more in the matrix a little bit better because there's lack of osmium tetroxide. Notice that the alcohol wash of the H&E tends to remove some of the lipids within the cells, causing them to have this kind of shriveled up appearance a little bit. They kind of collapse on themselves just a little bit. So that's an artifact of preparation from dehydrating this tissue with the alcohol. When stained with H&E, you can actually see some more details, like you can see the nuclei on these cells here. You may see some fibroblasts in the extracellular spaces or between the cells, in addition to the collagen fibers, which are these eosinophilic fibers. Now, some of your sections may have more of an appearance just like this when stained with H&E, and it really just depends on the quality of the slide, how it's been prepared and mounted. Histology is often more of an art than a science, and so in this case the integrity of the cell membranes has been compromised somewhat, so it's oftentimes tough to see all of the cellular structure here. Some of the cells have kind of degraded or collapsed on each other, but you can see lots of collagen fibers here in the matrix. So whether your slide looks like this or this really is going to depend on your particular slide. This is a section of reticular connective tissue, another type of adult connective tissue. You can find this in spleen and liver and some other structures. This particular section is from spleen that has been perfused, which washes out most of the red blood cells. Not all of the red blood cells have been removed, however you can see lots of those. These are the small disc-shaped cells here. But it leaves the fibrous stroma intact, and that stroma is composed of reticular connective tissue. Reticular connective tissue contains a large concentration of reticular fibers, more so than areolar tissues or other tissues which may also have them. Reticular fibers is actually a different type of collagen fiber. They're actually type 3 collagen fibers, but are usually just referred to as reticular fibers, which reflects an older system of nomenclature before the term collagen fibers was used, or before they were found to be different classes of collagen fibers. So this section has been silver stained, and silver stain shows the elastic fibers and stains them kind of a blue-black color. Notice they tend to be short, often branching, and they have this kind of mesh or net-like appearance, which is where reticulated fibers get their name. So this is reticular connective tissue. You can see a couple of blood vessels here that have taken up some background staining. So it's these fibers here that you want to look at. This is a section of thick skin. We've seen this before last week. Last week we focused on the epithelium, which is stratified squamous keratinized epithelium. This week we're actually going to focus on the connective tissue underneath the epithelium, which comprises the dermis. This type of connective tissue is classified as dense, irregular connective tissue. It's dense because, as you can see, the matrix is just packed with lots and lots of collagen fibers here, unlike the loose connective tissue, in which there are not nearly as many fibers and there's a lot more ground substance. This has less ground substance and a greater proportion of collagen fibers. So that's why it gets the name dense. It's irregular because you can see these collagen fibers, which are found in these thick bands here, have a very irregular shape to them, and they're going in every which direction. So they're irregularly arranged. That's why this type of tissue gets its name. So this is one of the places you can find dense irregular connective tissue, and the irregular arrangement of collagen fibers in multiple directions gives this type of tissue a lot of structural strength and lets it resist tension that's exerted in multiple directions. So it's suitable for places like skin that are going to be pushed and pulled from multiple directions to help keep everything together. You can see nuclei of various cells. Again, most of these are going to be fibroblasts. So these small basophilic structures here, those are the nuclei of mostly fibroblasts. The other type of dense connective tissue is called dense regular connective tissue, and it appears like the tissue you see in this section. This tissue gets its name because of the much more regular appearance of the collagen fibers, which are all layered together in a kind of laminar fashion. Again, there's very little extracellular space and very little ground substance, and the cells are squeezed in between these collagen fibers here, and so their nuclei often appear to be somewhat elongated. The arrangement of these fibers all in the same direction allows this type of tissue to resist tension only when pulled in a particular direction. So that gives it a lot of good tensile strength and is suitable for structures like tendons, where force is being exerted in a particular direction. Here's a section of connective tissue showing 4 fibroblasts here, as well as another cell. This cell here is called a mast cell. Mast cells are a type of immune cell. They are characterized by being fairly large cells. They have a large basophilic nucleus, and their cytoplasm contains lots of eosinophilic granules that you can see here. These granules contain histamine and other mediators of inflammation, so these cells help to regulate an inflammatory response to tissue injury or infection. These other cells, as we said, are fibroblasts, and again, mostly their nuclei are visible. This is actually a high magnification view, and you can see a little bit of the cytoplasm here towards the sides. But note that these cells are very skinny, and their cytoplasm is scant, giving a kind of tapered end or spindle shape appearance here. This is characteristic for the state of most fibroblasts in adult tissue that has been developed. And in this state, fibroblasts are said to be quiescent or inactive, in which they are only doing minimal amounts of cellular activity, maintaining the tissues. Here's connective tissue that is either developing or has been damaged, and so these fibroblasts have, many of them have transitioned from an inactive or quiescent state to an active state. And active fibroblasts have a different morphological appearance. Note that their cytoplasm is more full. The cells basically enlarge. They upregulate genes to help to repair or develop the matrix and are much more active, and so their cytoplasm increases volume to reflect that increased activity. Here's a higher magnification view of a couple of active fibroblasts right here, and so you can see the cytoplasm quite well. Oftentimes they have these sort of star-shaped appearances. This is said to be a stellate appearance or morphology. And so if you can see cytoplasm abundantly, then we classify those as active fibroblasts. So be able to distinguish active fibroblasts from the skinnier, more spindle-shaped inactive fibroblasts. Here's a section of tissue that is undergoing some kind of immunological challenge, probably a response to infection of some sort. And you can see there are lots of cells in this tissue here. These are mostly immune cells. There are two major types of cells you can see here. Most of these that have these large, deep basophilic staining nuclei, these are called plasma cells. Plasma cells are differentiated lymphocytes, which is a type of leukocyte that we're going to discuss later on. These cells are responsible for producing and secreting antibodies to help neutralize infectious agents. And here you can see many of them that have been recruited to this tissue because of a local immune response. The other major cell type you can see here are macrophages. And macrophages are large phagocytic cells. They are derived from monocytes, a different type of leukocyte that we're going to discuss later on. And they're just huge cells. You can see the nuclei here. The nuclear envelope stains very dark because of the large presence of heterochromatin around the nuclear envelope. And you can see their nucleoli. The rest of the nucleoplasm is quite pale. So this entire structure here is a nucleus, not a cell. The nuclei of macrophages are often so large that they're frequently confused with entire cells. Many macrophages have multiple nuclei as well. The actual cytoplasmic boundaries of macrophages, because of their irregular shape, can somewhat be difficult to differentiate from the background tissue, but you can see the borders of this macrophage here extends out quite a bit from the nucleus. Here's another macrophage, and it looks like their cytoplasm is fused a little bit, so it's a little bit difficult to see where one ends and another begins. So macrophages, here's another one, and then plasma cells are the main types of cells that you see on this section. And so you need to be able to identify macrophages, plasma cells, and mast cells that we showed you guys earlier. Just for review, this is a mast cell. This is what a mast cell looks like. Here you can see some inactive fibroblasts. So that wraps it up for generalized connective tissue. Now we're going to talk about a specialized type of connective tissue, which is cartilage. There are three main types of cartilage: Highland cartilage, elastic cartilage, and fibrocartilage. We're going to talk about Highland cartilage first. Highland cartilage is the most common type of cartilage. It differs from other connective tissues in that the matrix is solid and tends to have kind of a very homogeneous, glassy appearance. It also lacks, most types of cartilage have a different type of collagen, type 2 collagen rather than type 1 collagen, giving the matrix a much more smooth, homogeneous, or glassy appearance. So in this section we have trachea. Here you can see the epithelium of the trachea, which is composed of PCCE we talked about before. Here's some connective tissue underneath that. Here you can see some glandular tissue. We're going to look at those later on. This is actually a cartilaginous ring that is found in trachea that helps to reinforce the walls. That cartilaginous ring, it's a C-shaped ring. It's composed of hyaline cartilage. Hyaline cartilage is tough and helps to reinforce the walls of the trachea so that when you breathe in, the negative pressure doesn't cause the walls of the trachea to collapse on each other. Here's a higher magnification view of the cartilage. It's been stained with H&E, and you can see hyaline cartilage matrix is fairly basophilic. Some areas are more basophilic than others, and there are two distinct regions in the matrix we'll talk about in a minute that have different chemistries that make them react differently to the H&E stain. First I want to talk about the a section of the hyaline cartilage. So this is mature hyaline cartilage out here. This region is called the chondrogenic zone. This is called the chondrogenic zone because here's where you have chondrogenic cells that are constantly proliferating, producing new cells. So most of the cell growth occurs in this area in the chondrogenic zone by cells called chondroblasts. So you find chondroblasts in this region of the cartilage. The more mature cartilage out here contains chondrocytes. So chondroblasts initially lay down the cartilage, and then the cartilage is maintained by chondrocytes. Chondroblasts differentiate into chondrocytes. The chondrogenic zone contains a larger number of these chondroblasts in a smaller area. So you can see the cells are smaller and packed more closely together than those in the more mature matrix. Next to the chondrogenic zone, you can see this tissue right here. This structure is called the perichondrium, which means tissue that surrounds the cartilage. Perichondrium is actually composed of dense irregular connective tissue, so it's not cartilage itself. These eosinophilic fibers you're seeing here are the type 1 collagen fibers that are found in dense irregular connective tissue. So you can find fibroblasts in this tissue here. Here's a closer look at the mature cartilage. You can see the larger chondrocytes here that are found in these holes or depressions called lacunae. So chondroblasts and chondrocytes are found in lacunae because the matrix is solid, not liquid. The cells, after they lay down the matrix, they become trapped around with this solid matrix surrounding them. So the little holes or depressions where the cells live are called lacunae. So here is a single chondrocyte within a lacunae. Here's a lacunae with two chondrocytes. Sometimes you see multiple chondrocytes within a single lacunae after they've continued dividing, after they've left the chondrogenic zone. Notice that the coloration of the matrix differs depending on the distance away from the chondrocytes. So notice that the matrix immediately surrounding the chondrocytes is a more deeply basophilic color. This is called territorial matrix. This matrix is more acidic, and so it stains more basophilic, and that reflects the fact that it's less mature. As more mature matrix is laid down around it, this more mature matrix is farther apart and is more basic in color and composition, and so stains more eosinophilic. So as new matrix is laid down, this territorial matrix gets pushed out and new territorial matrix is laid down. As the older matrix matures, becomes less acidic, it's referred to as interterritorial matrix. So here you have interterritorial matrix and territorial matrix, which surrounds the chondrocytes. In this slide you can see lacunae, large lacunae with multiple chondrocytes within them. This one has at least 5 different chondrocytes you can see here. 1, 2, 3, 4, 5. Here you have 2 or 3. Here's 3. So as I said, some chondrocytes can continue to divide within lacunae through a process known as interstitial growth. Chondrocytes that are clustered together in these lacunae that are genetically identical, these are called isogynous groups or cell nests. So either of those words is interchangeable. So that's hyaline cartilage. Now let's take a look at another type of cartilage. This is a section from the epiglottis which contains elastic cartilage. Elastic cartilage is very similar to hyaline cartilage. Again, the matrix contains type 2 collagen instead of type 1 collagen, giving it the matrix also a very smooth, glassy appearance, just like with hyaline cartilage. A difference between elastic cartilage and hyaline cartilage is that elastic cartilage has a large number of elastic fibers. The elastic fibers, of course, give this type of cartilage greater elasticity, allowing it to stretch. So that's why you find it in the flexible epiglottis or the auricle of the ear, for instance, and a few other locations in the body. So this is elastic cartilage right here, surrounded by a perichondrium, again composed of dense irregular connective tissue. Here's a higher magnification view. Here you can see the perichondrium. Elastic cartilage also has a chondrogenic zone containing small chondroblasts within lacunae, as you can see here. Here the more mature is a more mature cartilage with chondrocytes in lacunae, and you can see that this particular tissue has been stained with an elastic stain that actually allows us to visualize these elastic fibers in more detail. So those are these kind of blue-black fibers. Elastic cartilage that hasn't been stained to show the elastic fibers looks very similar to hyaline cartilage. So if we show you elastic cartilage, it's always going to have been stained in this way. So if you can see these elastic fibers, you know it's elastic cartilage. So again, here you can see mature elastic cartilage. Here's the chondrogenic zone that contains chondroblasts, not chondrocytes. Notice the cells are smaller and packed closer together, and then again the perichondrium here. The last type of cartilage is called fibrocartilage. Fibrocartilage differs from hyaline and elastic cartilage in a couple of important ways. First off, instead of having type 2 collagen in the matrix, fibrocartilage has type 1 collagen, which means you can actually see the collagen strands in the matrix, which gives it a more kind of heterogeneous appearance rather than that smooth, glassy matrix you see in the other two types of cartilage. Another important difference is that fibrocartilage is not surrounded by a perichondrium, unlike the other two types of cartilage. There are two main places you can find fibrocartilage in the body. One is in the intervertebral discs, as you can see here. Notice that the fibrocartilage in this disc, the cells are arranged in a very regular laminar fashion. Another place you can find fibrocartilage is in the pubic symphysis. This is fibrocartilage from pubic symphysis, and notice it has a more irregular arrangement of cells, but it's still a type of fibrocartilage. In both types of cartilage, though, you can see the eosinophilic collagen fibers. Now, students have often told me in the past that sometimes they have trouble distinguishing between fibrocartilage and dense regular or irregular connective tissue. I think the best way that you know this is not dense connective tissue is that you look for the lacunae, because dense regular or dense irregular connective tissue has does not have a solid matrix, and so the cells are not found within the lacunae. Fibrocartilage does have a solid matrix, so these cells are found within lacunae, and you can see the lacunae surrounding these chondrocytes quite well in both of these slides. So if you see those, you know you're looking at fibrocartilage, not dense connective tissue. So that's what we have for you this week, and good luck studying.